You are not tired because you are old. You are tired because your cells have stopped making enough energy. And those two things are not the same. One is inevitable. The other is a biological process that has a name, a mechanism, and a growing body of evidence showing it can be slowed, and in some cases partially reversed. The fatigue most people over 60 accept as a permanent feature of their lives, the kind that sits on you even after a full night of sleep. The kind that makes a flight of stairs feel harder than it used to. The kind that pulls you toward the couch at 3 p.m. That fatigue, in many cases, is not your age. It is your mitochondria. And by the end of this video, you are going to understand exactly what is happening to them after 60, why it amplifies every other aging mechanism we have covered in this series, and the 5 foods that the research most consistently supports for restoring their function. But first, I need to tell you something that happened in a laboratory that most people outside the longevity research world have never heard about. Because it changes what exhaustion after 60 actually means. You learned in school that mitochondria are the powerhouse of the cell. That description is accurate. But it understates the scale of the problem when they start to fail. Mitochondria are present in virtually every cell in your body. Their primary job is to take the fuel from the food you eat, glucose, fatty acids, and convert it into a molecule called AP. Adenosine triphosphate. AP is the energy currency your cells use for everything. Every heartbeat, every thought, every muscle contraction, every immune response, every cellular repair process. 95% of the energy your body produces is generated by mitochondria. Not some of it. 95%. Now here is what happens when those mitochondria begin to age. A landmark review published in the journal Cell in 2023 identifying the 12 hallmarks of aging named mitochondrial dysfunction as one of the central mechanisms driving aging at the cellular level. Not a side effect of aging, a driver of it. The specific changes are well documented. Mitochondrial DNA, these organelles have their own DNA. Separate from your chromosomes, accumulates mutations over time from exposure to reactive oxygen species, the same oxidative byproducts that mitochondria generate in the process of making energy. The electron transport chain, the system of protein complexes inside the mitochondria that actually manufactures AP, becomes less efficient. It generates more oxidative stress and less energy output per unit of fuel. The result, documented in research published in PMC and Frontiers in Physiology, lowered oxidative capacity, reduced AP production, significant increase in reactive oxygen species, and diminished antioxidant defense. Your cells are running on less power, and the less power they have, the harder everything becomes. But there is a second mechanism happening simultaneously that makes all of this significantly worse. A molecule called NAD+. NAD+ nicotinamide adenine dinucleotide is a coenzyme that every cell in your body needs to run the mitochondrial energy cycle. Think of it as the essential catalyst without which the machinery cannot run. Without adequate NAD+, the electron transport chain stalls. AP production falls. Oxidative stress builds. NAD+ levels drop by roughly 50% between the age of 20 and 60. That figure comes from research published in the journal Cell in a landmark paper that linked this NAD+ decline to muscle deterioration, reduced mitochondrial function, and accelerated aging markers. 50%. By the time most people reach 60, they are running the same mitochondrial machinery they had at 30 on half the fuel. The research does not show this decline reversing on its own. In most people, without dietary or lifestyle intervention, it continues. Now add to that the relationship between mitochondrial dysfunction and everything else we have covered in this series. When mitochondria generate excess reactive oxygen species, those rows activate NF-κB, the master switch for inflammatory gene expression we identified in the inflammaging video. Dysfunctional mitochondria release signals called DAMPs, damage-associated molecular patterns that trigger the same immune responses as the SASP from zombie cells. Reduced AP production impairs the autophagy process that is supposed to clear senescent cells and maintain the gut lining. Mitochondrial decline does not sit beside the other mechanisms in this series. It amplifies all of them. This is why fatigue after 60 is rarely just fatigue. It is the visible surface of a much deeper failure. There is one more piece of this you need to understand before we get to the foods. Because without it, you will not fully appreciate why one of the 5 foods is the most exciting finding in mitochondrial research published in the last 2 years. Mitophagy. Mitophagy is your body's quality control system for mitochondria. It is a selective form of autophagy, the cellular recycling process we covered in Video 1, specifically designed to identify dysfunctional mitochondria and clear them out so that healthier ones can replace them. In a young body, mitophagy runs efficiently. Damaged mitochondria are removed. New ones are created through a process called mitochondrial biogenesis. The population of mitochondria stays functional. In aging, mitophagy slows. Dysfunctional mitochondria accumulate rather than being cleared. They continue generating oxidative stress and inflammatory signals long past the point when a well-functioning system would have removed them. The pile of broken-down cars on the freeway, the metaphor we used for zombie cells in Video 4, applies here too. But instead of zombie cells, it is zombie mitochondria. Still occupying space, still generating toxic byproducts, and no longer being cleared away. The solution, researchers now understand, is not just to supply the mitochondria with better fuel. It is to restore the clearance system that removes the damaged ones and creates new, healthy replacements. And a compound derived from pomegranate through a process that goes through your gut bacteria turns out to be the most significant natural activator of that system yet tested in human clinical trials. Before I show you the 5 foods, I want to tell you about a woman I will call Carol. Carol is 69. She had been a competitive recreational cyclist until she was 65. Not professional, but serious. Long rides on weekends. Consistent training through the week. Around 66, something changed. The rides that used to feel demanding began to feel impossible. Not harder impossible in a different way, like her body simply could not access the energy she knew should be there. Her doctor ran standard blood tests. Everything came back normal. She was told she was in good health for her age. Carol found her way to this series through a friend who sent her Video 1. She made changes to her diet over 3 months, specifically targeting her mitochondrial function. At 9 months, she was back on long rides, not racing, but riding. She wrote, "I spent 2 years thinking I had just gotten old. It turns out I had just gotten hungry. My cells were hungry. I did not know you could feed mitochondria. I cannot promise you Carol's experience. Every person is different. These dietary changes are not medical treatment. Please always speak with your doctor before changing your health routine." What Carol changed and what the research consistently points to comes down to 5 things. Food, number 1. Fatty fish. Salmon, sardines, mackerel, and herring have appeared in every video in this series. This is the last time I need to explain why, because by now, you understand the common mechanism. For mitochondrial function specifically, the omega-3 fatty acids EPA and DA are directly incorporated into the inner mitochondrial membrane. The fluidity and structural integrity of that membrane determines how efficiently the electron transport chain can operate. When the membrane is enriched with omega-3 fatty acids, electron transfer is more efficient and AP production is higher. When it is dominated by saturated fats and processed vegetable oils, the chain becomes sluggish. Research published in Frontiers in Aging shows that omega-3 supplementation is associated with improved mitochondrial coupling efficiency and reduced mitochondrial oxidative stress in aging muscle tissue. 2 to 3 servings per week. Sardines and mackerel are the most concentrated CoQ10 sources among fish. Mackerel provides approximately 6.75 milligrams of CoQ10 per 100 grams. Sardines approximately 6.4 milligrams, meaning fatty fish is simultaneously providing the structural material for the mitochondrial membrane and the coenzyme that runs the electron transport chain. Food number 2. Organ meats. This is the food most people would not expect on this list, and it is the one with the highest CoQ10 content of any dietary source. CoQ10, coenzyme Q10, sits directly inside the mitochondrial electron transport chain. It shuttles electrons between protein complexes in the chain, and without adequate CoQ10, that chain becomes inefficient and generates more oxidative stress as a byproduct of energy production. CoQ10 is naturally produced by the body, but its production declines with age. And critically, if you are taking a statin medication for cholesterol, which many adults over 60 are, statins inhibit the same metabolic pathway used to synthesize both cholesterol and CoQ10. Statin use without CoQ10 replacement is one of the most underappreciated contributors to the fatigue experienced by older adults on these medications. The richest dietary sources: beef heart provides approximately 11.3 milligrams of CoQ10 per 100 grams. Chicken liver provides approximately 11.6 milligrams. Beef liver provides 3.9 milligrams. These are not exotic foods in the context of human history. Organ meats were a dietary staple for millennia. If organ meats are not accessible or appealing, chicken and beef also contain meaningful CoQ10, approximately 1.4 and 3.1 milligrams per 100 grams respectively. And sardines and mackerel, as noted, provide 6 to 7 milligrams. One important precision note: the dietary amounts of CoQ10 from food are substantially lower than the 100 to 300 milligram doses used in clinical supplementation trials. Food-based CoQ10 is a meaningful daily contributor, but if your doctor has identified mitochondrial dysfunction or you are on statins, a conversation about CoQ10 supplementation is worth having. Food number 3. Pomegranate and dark berries. This is the most exciting food on this list in terms of recent human clinical evidence. Pomegranate contains compounds called elagitannins. When elagitannins reach your colon, certain gut bacteria, and notably, only those people who have the right gut microbiome composition, convert them into a compound called urolithin A. And urolithin A is, at this moment, the most rigorously studied natural activator of mitophagy in human clinical trials. A 2025 randomized, double-blind, placebo-controlled trial published in Nature Aging: The Mitoimmune Study found that 4 weeks of urolithin A supplementation in healthy middle-aged adults produced augmented mitochondrial biogenesis in immune cells, increased levels of naive CD8 T cells, the fresh, adaptable immune cells that decline with age, and enhanced the metabolic profile of those cells, including increased capacity for fatty acid and amino acid oxidation as fuel. The mechanism: urolithin A stimulates mitophagy, clearing out damaged mitochondria, and simultaneously activates PGC1 alpha, the master regulator of mitochondrial biogenesis. More clearance of damaged mitochondria, more creation of new ones. The quality control system restored. 2 important caveats. First, the Mitoimmune trial used 1,000 milligrams of supplemental urolithin A daily, a concentration that dietary pomegranate alone cannot deliver. Eating pomegranate supports urolithin A production in people with the right gut bacteria, but cannot replicate the clinical trial dose. If you want the direct compound at therapeutic levels, supplemental urolithin A is available. Second, not everyone produces urolithin A from dietary elagitannins at all. The conversion depends on having specific gut bacteria, which connects directly to video 6. People with depleted gut microbiomes may produce little or none. This is one of the strongest arguments in this entire series for maintaining gut microbiome diversity. It is not just about memory and mood. It is about whether you can activate your own mitochondrial quality control system. Practical target: half a pomegranate, 4 ounces of pomegranate juice without added sugar, or a daily serving of dark berries, blueberries, and blackberries also contain elagitannins and related compounds. Consistent dietary intake is a meaningful starting point, with the gut microbiome doing the conversion work. Food number 4. Green tea. Egg C. epigallocatechin gallet has appeared in multiple previous videos in this series for its autophagy activating and SASP reducing properties. For mitochondria specifically, it adds a third mechanism. Egg C activates AMP activated protein kinase, which is often called the energy sensor of the cell. When cellular energy is low, AMP activates a cascade that includes PGC1 alpha, the same mitochondrial biogenesis master switch activated by urolithin A, and directly stimulates the creation of new mitochondria. Research published in journals including Molecular Nutrition and Food Research has found that EC reduces mitochondrial oxidative stress, supports mitochondrial membrane potential, and is associated with improved energy metabolism in aging tissue. 2 cups of plain green tea daily. Decaffeinated retains meaningful egg CG content. Brewing at 80 degrees Celsius rather than boiling water preserves more of the active compounds. Food number 5. Magnesium-rich foods. This is the one most people would not guess, and it may be the most immediately impactful for fatigue specifically. The energy molecule mitochondria produce does not function in its free form inside cells. It functions as magnesium ATP. Magnesium must bind to ATP to make it biologically active. Without adequate magnesium, even the AP your mitochondria produce cannot be used efficiently by your cells. Research estimates that approximately 50 to 60% of adults in Western countries are deficient in magnesium. In adults over 60, the percentage is higher because magnesium absorption from the gut decreases with age, and because many medications common in older adults, including certain diuretics, PPIs, and diabetes medications, deplete magnesium further. A systematic review published in Nutrients in 2023 found that magnesium deficiency was independently associated with increased fatigue, muscle weakness, and higher inflammatory markers, including CARP and IL-6, directly connecting the magnesium-AP relationship to the inflammaging fire we covered in video 5. The most bioavailable dietary sources, pumpkin seeds, are the richest single source, providing approximately 150 milligrams per 28 gram serving. Dark leafy grain spinach and Swiss chard are excellent sources and also appeared in video 1 for their prebiotic fiber. Dark chocolate at 70% cocoa or higher provides approximately 64 milligrams per 28 grams. Legumes, black beans, edamame, lentils provide 60 to 120 milligrams per serving. Target: 320 to 420 milligrams of dietary magnesium daily, depending on sex. Most adults over 60 are significantly below this. Consistent dietary magnesium is often the simplest, fastest intervention for the low-grade fatigue that does not respond to sleep. Let me give you the larger frame before we close. In 7 videos, this series has traced a single thread. The gut microbiome declined. The vagus nerve signal weakened. Gut bacteria drove memory loss through the hippocampus. Zombie cells filled tissues with inflammatory chemicals. A low-grade fire called inflammaging burned through organs. Bacteria stopped producing BDNF, and cognitive sharpness drifted. And underneath all of it, amplifying every single mechanism, the cellular engines were running on insufficient fuel. Less NAD+, less CoQ10, less efficient electron transport, slower mitophagy. the accumulation of damaged mitochondria generating more oxidative stress and more inflammatory signals, feeding the SASP, feeding the inflammaging fire, feeding the gut dysbiosis. The mitochondria are not the only story in aging, but they are the energy source that makes every other story worse. And what is extraordinary about where the research now stands is this: 5 of the 7 mechanisms in this series are addressable through the same dietary pattern. The same fatty fish, the same dark berries, the same leafy greens, the same olive oil, the same fermented foods. There is not a different protocol for each problem. There is one underlying biology, and it responds consistently to a small number of specific inputs. Here is what I want you to carry out of today. The exhaustion you have been attributing to age is, at least in part, a fuel problem. Your cells are not generating enough energy because the system that makes that energy has been quietly degrading for years without a symptom you would recognize as a mitochondrial problem. It is not a character flaw. It is not weakness. It is a biological process with a biological response. You can feed your mitochondria. You can support the quality control system that clears out the damaged ones. You can supply the coenzymes the electron transport chain needs to run efficiently. And none of it requires a prescription. So here is your one action for today. At your next meal, eat a handful of pumpkin seeds, approximately 28 grams. That is 1 serving, roughly 150 milligrams of magnesium, and the simplest first step toward giving your mitochondria the mineral they need to make the energy you are already producing biologically active. 1 handful. 1 step. Your cells are not running out of time. They are running low on fuel. And you can start today. Quick recap. Mitochondrial dysfunction is named as one of the 12 hallmarks of aging in a 2023 Cell review. NAD+ drops by roughly 50% between age 20 and 60, reducing the fuel available to the electron transport chain. Mitophagy, the clearance of damaged mitochondria, slows with age, allowing dysfunctional mitochondria to accumulate and amplify the inflammatory signals driving every other mechanism in this series. 5 foods with the strongest research support: fatty fish, for omega-3 enrichment of the mitochondrial membrane and dietary CoQ10. Organ meats for the highest dietary CoQ10 concentrations essential if you are on statin medication. Pomegranate and dark berries for elagitannins that gut bacteria convert to urolithin A, the most clinically validated natural activator of mitophagy, demonstrated in a 2025 Nature Aging randomized controlled trial. Green tea for EGCG, which activates AMP and stimulates mitochondrial biogenesis. And magnesium-rich foods: pumpkin seeds, leafy greens, dark chocolate, legumes because API cannot function in cells without magnesium, and deficiency is widespread in adults over 60. This is the 7th video in this series. We have now covered the 7 core mechanisms of biological aging: the gut microbiome, the vagus nerve, the memory pathway, zombie cells, inflammaging, cognitive bacteria, and mitochondrial energy. The next video is going to do something different. Instead of adding another mechanism, it is going to show you how all 7 of them connect into a single daily protocol, the minimum effective dose of dietary changes that addresses all of the biology we have covered in one approach. It is the video that makes the series actionable. Subscribe button is just below if you want to see it. And if someone in your life has been told that fatigue after 60 is just aging, please share this series. The cells are hungry, not tired. Always speak with your doctor before making changes to your diet or health routine. Everything here is for educational purposes only. Take care of yourself. And I will see you in the next one.